This video will cover the cells and organs that make up the immune system. All blood cells arise from hematopoietic stem cells, which are self-renewing and differentiate into various cell types. This process is called hematopoiesis. During early hematopoiesis, a multipotent stem cell differentiates into either a lymphoid progenitor cell or a myeloid progenitor cell. Progenitor cells are committed to a specific cell lineage and lose the capacity for self-renewal. Lymphoid progenitor cells give rise to B, T, and natural killer cells. Myeloid progenitor cells generate progenitors for red blood cells, various white blood cells, and megakaryocytes. Hematopoiesis is regulated at the genetic level, with genes like GATA2 being an example of this regulation. The cells of the immune system are fundamental to defense against pathogens. Lymphocytes are central to adaptive immunity, providing diversity, specificity, memory, and self-non-self recognition. Other white blood cells engulf and destroy microorganisms, present antigens, and secrete cytokines. Lymphocytes constitute a large portion of white blood cells and circulate through the blood and lymph. They are subdivided into B cells, T cells, and natural killer cells based on function and membrane components. B lymphocytes, or B cells, mature in the bone marrow in mammals. Mature B cells display membrane-bound immunoglobulin, which acts as a B cell receptor for antigens. This binding leads to rapid division and differentiation into plasma cells and memory B cells. The B cell receptor is an important molecule on the surface of B lymphocytes. T lymphocytes, or T cells, mature in the thymus. They express a T cell receptor that recognizes antigens bound to major histocompatibility complex, or MHC, proteins on cell membranes. T cells are categorized into T helper, T cytotoxic, and T regulatory cells. T helper cells express the CD4 glycoprotein, while T cytotoxic cells express CD8. Natural killer, or NK cells, are lymphocytes that exhibit cytotoxicity against tumor cells and virus-infected cells without prior immunization. They constitute 5% to 10% of peripheral blood lymphocytes. NK cells recognize target cells in two ways. First, through a reduction in class I MHC molecules using a killer cell inhibitory receptor. Second, through antibody-dependent cell-mediated cytotoxicity, binding to antibodies on target cells via CD16. When an NK cell's inhibitory receptor binds to MHC class I on a normal cell, no lysis occurs. If the NK cell's receptor does not bind to MHC class I, as often happens with virus-infected or tumor cells, lysis occurs. The mononuclear phagocytic system includes monocytes in the blood and macrophages in the tissues. Phagocytosis is an initial activating stimulus for these cells. Macrophage activity is enhanced by cytokines, inflammatory mediators, and bacterial components. Activated macrophages secrete cytokines like IL-1 and TNF-alpha, complement proteins, and hydrolytic enzymes. Macrophages are dispersed throughout the body. Fixed macrophages reside in specific tissues and include alveolar macrophages, Kupffer cells, and microglial cells. Free macrophages are motile. Granulocytic cells are classified as neutrophils, eosinophils, or basophils. Neutrophils are the first responders to inflammation and are active phagocytic cells that extravasate into tissues. Neutrophils are highly effective at killing ingested microorganisms, utilizing lytic enzymes and bactericidal substances within their granules. Eosinophils are motile phagocytic cells primarily involved in defending against parasitic organisms. Basophils are non-phagocytic granulocytes that release pharmacologically active substances, playing a role in allergic responses. Mast cells are undifferentiated until they enter tissues and are also important in allergies. Dendritic cells, or DCs, are covered with long membrane extensions. Most mature DCs function as potent antigen-presenting cells, or APCs, to T helper cells, expressing high levels of MHC and B7 co-stimulatory molecules. Follicular dendritic cells do not arise in the bone marrow and do not function as APCs for T helper cell activation. They express high levels of antibody receptors, allowing them to bind antigen-antibody complexes. The immune system is organized into specialized organs. Primary lymphoid organs, like the thymus and bone marrow, are where lymphocyte maturation occurs. Secondary lymphoid organs, such as lymph nodes and spleen, trap antigen and facilitate interactions with mature lymphocytes.